In the previous segment, we saw the data that led Mendel to conclude that all of the traits that he was studying, those 7 traits, were independent of each other. We also walked through the demonstration of his explanation, meaning we saw how his explanation leads to the observations that he made. So we now have this, what Mendel is saying for the 7 traits, and again, only those 7 traits so far that he's been studying, that the genetics are particulate. 2 particles determine each trait, 1 comes from each parent, 1 of them dominates, and all 7 are independent of each other. Now, remember though, the data that he used to generate the hypothesis of independence cannot then be used to support the hypothesis of independence. And again, the reason is because, of course, the data that generated the hypothesis fit the hypothesis. That's the whole point. But now what he has to do is like we saw before. What's he going to do to test it? How does he have to test this hypothesis? And like we saw before, he's got to predict the result of an experiment that he had not performed yet. So, can he use the same principle here? Can he use essentially the same kind of experiment here? And the answer is yes, that's what he's going to do. He's now going to predict what would happen if he does a test cross. Now remember the test cross. The test cross is crossing the F1 with its recessive parent. Okay, if we go back all the way up here, we're talking about crossing this individual here with the recessive parent. Okay, as always, do we know the genotypes of the 2 parents for this cross? And the answer is yes. We know the genotype of the offspring. It's the dihybrid, big R, little r, big Y, little y, and we know the genotype of the recessive parent, little r, little y, little y. So, in this case then, we do know the genotypes of the parents, so we can now predict what we would expect to see. So here's the point then, what kind of gametes can this F1 make? Because we have to draw a Punnett square, or if we're going to use that technique, we have to draw a Punnett square in order to be able to determine what's going to happen. So knowing that, can we determine what the genotype is of the gametes made by the F1? And the answer is yes, they're right here. Here's the F1, here are the kind of gametes it can make. Now remember, the gametes are made by 1 R, 1 Y put into each gamete. Therefore, since I have 2 R's, big R, little r, and big Y, little y for the Y's, I can get these 4 possible gametes. We've been through that. What about the little r, little y, little y? Well, again, we've been through that as well. It can only make 1 little r, 1 little y in the gamete. So the gametes have to be coming out of this parent, have to be little r, little y. That's the only thing that can happen. There can be no randomness associated with that. Okay? So now let's do a Punnett square. As we saw before with the Punnett square, you have to write out all of the gametes that can be made. Here's the gametes from one, here's the gametes from the other. So our result is not going to be this. Our result is going to be these 4 crossed with this, and that's not on this page. So we have to draw that out. So if we draw that out, here's our box. Here are the gametes that can be made by the F1, because it's big R, big Y, little y. Okay? Here's the only kind of gamete that can be made by the recessive parent. Okay, so that means this. If this is the male, then all the sperm are little r, little y, and here are all the possible eggs. Or vice versa. Here are all the possible sperm, and the only eggs that can be produced are little r, little y. All right, so if we combine these like we do normally, we can also get the phenotypes as well as the genotypes. For example, this is the sperm and this is the egg. This sperm fertilizes it, gets big R, big Y, little y. Big R, little r makes it round. Big Y, little y makes it yellow. So that would be round yellow. Okay, in this case then, what would be this? Well, you have big R, little y crossed little r, little y, which gives us big R, little y, little y, which would be round and green. This one is going to end up being wrinkled because little r is the result, and a big Y gives us yellow. And then finally we get all little, which is going to give us wrinkled green. Now notice the way I did this. I didn't repeat this little r, little y 4 times because I don't need to. There's no point in repeating this over and over again. Or technically I should, in a sense, if we're doing this the way I've mentioned it, because it could be the left hand little r and the left hand little y, that's the first. It could be the left hand little r and the right hand little y, that's the outers. Notice we're doing FOIL again. The middle ones, or the end ones, but it doesn't matter, they're always the same. So I don't have to repeat the same thing over and over again because I'm just going to get those same things out, and means it's not going to change the ratios in any way. So therefore, if we do this experiment, we expect this relationship, a 1 to 1 to 1 to 1 ratio. In other words, we expect equal numbers of each of these different phenotypes in the resulting experiment. So again, if what Mendel said is correct, if his hypothesis is correct, we would expect equal numbers of each offspring from this cross. All right, so here are the results that he actually got. In his experiment, he did two different types of crosses. One where he had the female plant was the round yellow and the male plant was the wrinkled green, and the other where the male was the round yellow and the female was wrinkled green. And what he has then is this. He takes this round yellow F1, crosses it with the recessive parent, and what he got out of that experiment was 31 round yellow, 26 round green, 27 wrinkled yellow, and 26 wrinkled green. Okay, so just think about that for a moment and compare that to what we just predicted. Now, in the other experiment where he switched the genders of the two different parents, he got 24 round yellow in the offspring, 25 round green, 22 wrinkled yellow, and 27 wrinkled green. Okay, think about this. Do these results here support, contradict, or say nothing about Mendel's predictions from his principle of independent assortment? Okay, well, from this bit that we saw before, we're expecting equal numbers, but are we expecting exactly equal numbers? Well, just like we saw before, no, we're not, because there's randomness associated with this gamete that gets chosen, not this one. This one's always got to be little r, little y, but there's an equal probability of choosing any of these. In other words, it's like sort of coming up with a four-sided die, if you will, and you roll the die, and if it comes up 1, it's this, 2, it's that, 3, it's that, 4 is that. It's possible, however, if you do that, you roll 1 every single time, in which case they're all going to be round yellow, which is going to move you away from 1 to 1. So you don't expect to get exactly this 1 to 1 to 1 to 1 ratio. Okay, so he didn't get exactly that, but he got almost equal numbers in each case. Okay, so that without question supports. Don't be afraid to say it supports. We're not saying this. What do these results prove? What do they prove? Well, nothing. They prove nothing. But again, we're not trying to prove anything. All we're simply saying is, do these results support a conclusion or do they not? And these results, without question, do support this conclusion. Now we can do statistics on this to double check to see how likely it is that we got this big a deviation, 31 and 26, from exactly equal numbers by random chance. If we do that, the probability is very low. So that's why we say it supports. It without question does support that conclusion. It's what you're predicting. But they don't prove anything. Is there more here, though? Is there anything else that we can conclude from these data? And whenever you see a data set, that's what you always want to do. Try to get as much information out of the data set as you possibly can. And don't be afraid of it. Don't be shy and say, oh, well, I can't tell anything because I need more study. Well, of course we need more study. We always need more study. More study is always better. But there are times when we're so certain of something that more study is really going to be useless. In this case, yeah, more study is probably needed, but we've done that. But the fact of the matter is that's not what we're asking. We're not asking, should we do more studies? We're asking, what does this study suggest? And this study supports this conclusion of independent assortment because we got what Mendel predicted based on the prediction of independence. Okay, but what else can we conclude? Well, here's the question. Does it matter which parent is the round yellow one? Does it matter which sex is the round yellow parent? And the answer is no, because we got essentially equal numbers when the male was the round yellow and when the female was the round yellow. It didn't make any difference. So that's why Mendel did this. He was trying to see, is there any relationship to the sex of the plant? And the answer is no. So what really this shows is 3 things are independent. The seed shape, the seed color, and the gender are independent of each other. Okay, so this is just a summary of what we said. This supports the principle of independent assortment because it fits the prediction. It doesn't prove anything. So again, we're not in the business of proof, but again, it doesn't make a difference which parent contributes which trait. They're still independent. Okay, again, the question then is, why is it not exactly 1 to 1 to 1 to 1? Why is it not equal numbers of each? Well, like we said before, the issue is this. Which of these gametes gets chosen to be fertilized by this gamete or vice versa is random. This one is totally random. Okay, so that's why it is we can't we don't expect to get exactly equal numbers, a quarter of each, because this has got some randomness associated with it. So there's a lot more to Mendel's paper that I'm not going to really talk about. I want to leave the paper now and talk a little bit about the history of what is going on, because this is really astonishing. Mendel tried to get people to understand what he was saying, and they understood it, but they didn't believe him. And again, the reason they didn't believe him, it appears to be because they were so entrenched in this theory that was incorrect, things like this generative fluid theory. And Mendel was saying, look, I don't believe I've made a mistake. I don't believe any of my results are wrong. And they imply that the generative fluid idea must not be true. Now, he can't get really anybody to believe that in his lifetime. So unfortunately, he dies, if I remember correctly, around 1865-ish, something like that. Then at the turn of the 20th century, okay, so 35 years later, 3 different individuals studying genetics stumble on the same discovery that he made 35 years or more earlier than that, because he made the discovery around 1835. Luckily, they saw it, and they recognized that he had priority, even though he was dead, and so they gave him credit, which is why we know his name. Now, at about that same time that Mendel was rediscovered, this individual, a guy by the name of Walter Sutton, working at Columbia University, made another discovery, which was absolutely critical in nailing down the reality of Mendel's results. And he was studying, oddly enough, this grasshopper. He wasn't studying people. He wasn't a medical doctor or anything like that. He was studying this grasshopper. Why? Well, it's a plains lubber grasshopper, Brachystola magna, or if you wish, Brachystola magna. And this creature is a pest. So, I mean, it has economic importance and so on. But he was studying it because the cells were easy to obtain and the chromosomes inside those cells were easy to deal with. Now, the thing you got to remember is this. Chromosomes were not discovered until after Mendel died. Mendel knew nothing about chromosomes. In fact, he barely knew anything about cells. It's not even clear he knew what cells were. And this guy was one of the first people ever to study chromosomes. Here are some of his earliest drawings of the chromosomes that he was able to obtain from this individual, this type of creature. And as he's studying these chromosomes, he's also studying what they're doing and how they're operating at the time the cells are dividing. And he finds in the tissues that are the generative tissues of this creature, not the generative fluid, but the generative tissues, meaning these are the tissues that give rise to sperm and egg, he discovers something very interesting. He says, okay, look, all my studies, I find that chromosomes come in pairs. The offspring get one of each pair from each parent. And the pairs separate in the process of meiosis. This is essentially the discovery of meiosis. And they segregate into different gametes. Okay, now think about that for a moment. They come in pairs, the chromosomes come in pairs, and at the time that they make gametes, the paired chromosomes separate. And one goes into one gamete, another goes into another gamete. All right, so he then says, in his own words, this: the association of paternal and maternal chromosomes in pairs and their subsequent separation during the reduction division may constitute the physical basis of the Mendelian law of heredity. All right, let me translate that. The association of dad's and mom's chromosomes in pairs. Remember, Mendel said the particles are coming in pairs. They're their pairs. And those particles, you get one from each parent, which means that when you are a parent, you have to take one of each chromosome or one of each particle and give it to each gamete and have those go off independently of each other. And there's subsequent separation. That's exactly what I just described. During the reduction division, now that's what he called meiosis 1, which we studied in laboratory, is where the chromosomes separate and the cells go from diploid to haploid. They may constitute, that observation may constitute the physical basis of Mendelian law of heredity. Meaning his chromosomes are acting like Mendel's particles. They're acting just like Mendel's particles. And they're particles themselves. So this is what's absolutely astonishing. Mendel, 35 years before the discovery of chromosomes, he had no idea what chromosomes were. Just by looking at numbers of individuals born from particular crosses, just looking at the numbers of individuals born from crosses, predicted the existence of meiosis. That is badass. All right, so let me look at this diagrammatically. Let's talk about what Walter Sutton was actually saying in terms of the diagrams. Now, we're talking about the same sort of issues that we were talking about with Mendel. We're going to combine the two. Now, here's a cell, and it's got 4 chromosomes in it, and this big blue chromosome is carrying R. And this chromosome here is also carrying R, but it's red. Now, in the way this picture is made, they're showing homologous chromosomes in different colors. The chromosomes that came from one parent, say the mother, are blue, and the chromosomes that came from the father are red. Okay? So the mom is going to give one big chromosome and one little chromosome. And on one big chromosome, it's carrying a gene for R. Now, remember we talked about what genes are. Genes are the particles, but they're not the entire chromosome. They're just part of the chromosome. So there would be the gene right there for big R on the chromosome that came from mom. So mom is giving, has a big R, or its mom had a big R. Its dad has a little r. Okay, so it's got the chromosome that came from dad. Okay, now these chromosomes, note, are the same size. Their centromeres are in the same location, and they're both carrying the same gene. They're both carrying the seed-shaped gene, R. That makes them homologous by the definition that we used earlier. The small chromosomes here are carrying Y, and they're homologous as well by the same definition. Okay, now, let's look at this. During mitosis, these chromosomes all line up in the metaphase plate, but this cell is not going to go through mitosis. This cell is what we call a gamete mother cell. The gamete mother cell is going to go through meiosis. Remember, meiosis has 2 sets of divisions. That comes from the laboratory, so if you haven't done the lab yet, you'll start to nail that down as we go. But when we go to my metaphase of meiosis 1, the very first division of meiosis 1, the chromosomes are side by side. The homologous pairs are side by side. And that's different than what happens in mitosis. Mitosis, they're all in a line, and you took pictures of that, of the chromosomes that you've cut out. If you haven't done that, you will. Okay, now, if this is the way that they're organized, where the mother's chromosomes are on one side, the father's chromosomes are on the other, then during anaphase, these 2 chromosomes will go into its own cell, and these 2 chromosomes will go into its own cell. In which case then, this cell will end up with big R, big Y, and this cell will end up with little r, little y. Okay, so that's would be how they segregate. Then in meiosis 2, which is identical to mitosis, they simply break the 2 sister chromatids apart, become daughter chromosomes, and we end up then with each of these being the same. Okay? Now, they're not showing crossing over in this picture because crossing over in this case doesn't matter. The reason is because these genes, R and Y, are on different chromosomes. And that's why they're independent. If they were on the same chromosome, they wouldn't be independent because they'd be passed together with issues with recombination. We'll talk about that later. But the point is that these are on different chromosomes, therefore they're independent. The R's and the Y's are independent of each other. But notice how this works. The 2 chromosomes that are homologous are separated from each other. Okay, so this big chromosome and this big chromosome go in opposite directions, which makes perfect sense. If these 2 chromosomes went to one gamete, then this gamete would have 2 genes for the seed shape and none for seed color. And this one would have 2 genes for seed color and none for seed shape. That doesn't make sense. The offspring has to have seed color and seed shape. And this parent is only going to give one of each. So that means the homologous chromosomes have to go in this pattern where the big and the little go to one and the big and the little go to the other. Okay, so that's one way that can happen, and when we get that, then that means all the gametes here are big R, big Y. These gametes are little r, little y. But it's equally likely, and here's why they're independent, it's equally likely that the chromosomes, when they get into metaphase 1 of meiosis, end up in this configuration, where this little chromosome from the father segregates with the mother's big chromosome, in which case these gametes are going to carry big R, little y. And of course these gametes are going to be carrying little r, big Y, because dad's little r chromosome is going to be going with mom's big Y chromosome. And then again, go through mitosis, these end up being the same as this, and these end up being the same as that. So that then tells us that if these are independent on different chromosomes, we're going to end up with equal numbers, 1/4 of each possible combination. So when we saw that earlier, and when Mendel was predicting that, that was meiosis. That's exactly what meiosis does. It separates those things out, which is why it is that Mendel was so astonishing in predicting the existence of meiosis simply by looking at the numbers of offspring from these reproduction events that he was studying.